Hey, Darren.
Morning, Andrew. How are you?
Thanks for inviting me. This should be fun.
Yeah, looking forward to it. Thanks for agreeing.
Good morning, everybody.
Morning, Gabrielle.
It's nice to see you all. We have people coming in from all over the place, which is always fun. We're going to go ahead and get started. So good morning again, everybody. Welcome to another episode of Wildland Stories. My name is Gabrielle Harden, and I am joined today by my co-host, Darren McAvoy.
Good morning.
Today, we're going to be digging in to the world of soils. We're going to start out with some really basic stuff, and then we're really going to grill our guests with the questions that you guys submitted. They were a little bit more in depth, so I think you'll enjoy that. Our guest today is Andrew Kulmatiski. He's an associate professor here at Utah State University.
Good job with my name.
I'm sorry?
Good job with my name.
Oh, thanks. I had
Most
to sound it.
most people have trouble.
I practiced a few times, and I was just hoping I would get it right. He got his master's of forest science at Yale School of the Environment and his PhD in biology here at Utah State University. Much of his research focuses on examining the mechanisms through which large-scale human activities affect plant-soil interactions. So if you're ready, Andrew, we're just going to dive right in and start harassing you with some questions.
Yeah.
Okay, so we're going to start off super simple. I did want to tell everybody first, all of our audience, if you have a question during the entire webinar, please put it in the Q&A box. It's super hard for me to sift through that chat box to find your questions. I am the host and the tech, so if you put them in the Q&A, it's a lot easier for me to find them. Okay, so what is soil science?
Okay. I have a history of giving obtuse answers, so I'm going to try to give some very simple answers first, and then I'll go into the obtuse answers. So soil science is the study of soils. So there's a lot of different aspects to it. There are some who focus on soil physical properties. I used to do construction inspection, and so we looked only at the physical properties of soils. There are others who focus more at how soils affect plant growth and soil nutrient cycling. So there's a lot of different aspects to it. I think I don't really think of there being a real hard definition for it. In my work, I actually look at soils as sort of a medium on which plants grow. So I'm not a strict soil scientist. I don't study only aspects of soils. I look at sort of from a whole ecosystem level, how do plants and soils interact.
Andrew, do you consider yourself a soil scientist? I work with you, and I think of you as more of a soil ecologist. I've heard you present, and I know a little bit about your work. It seems like it's in that space between soils and plants where you focus, especially compared to other soil scientists that I work with.
Yeah, I'd say I'm more of an ecologist, but my work is all based in the soils. It's kind of like I grew up in New York, and then I went to school in Maine, and when I told people I was from New England, they said, "You're not from New England. New York's not in New England." So when you talk to a soil scientist who only strictly studies soils, they'll say, "Oh, he's not a soil scientist." But all of my research is based out of the soils. So
thanks for that understanding.
So when and how did your passion and interest for soils start?
I was pretty lucky in that in high school, I knew what I was going to do. I took biology in high school, and we went for months, even back in the 80s and 90s when I was going to school. We learned about the genes that control photosynthesis and all the molecular mechanisms of how photosynthesis works. We covered that for months. And then we got to soils, and soils was just an arrow. There's a black box on which plants grow, and everything then just loops through there, and nutrients come back up. And I was like, hold on, wait. I think there's probably more going on in that other half of the ecosystem. And so from that time, I thought, wow, there's got to be a lot to learn about what's going on underground.
That is funny, because I do remember they just put like a little arrow, and they just like, this is where it grows from, and then that's it. That's all they had to say about that.
Yeah. So we have a lot of job security in soil science. We have a lot to learn.
So do you have an undergraduate degree in soils? Is that what you studied right off the bat?
I had, so because, it wasn't very well developed. I went to a liberal arts college. I got a biology degree with a chemistry minor. And so there wasn't there wasn't an option to study soils at the school I went to. I mean, you have to get to a pretty big, land grant university to get the soils department.
So correct me if I'm wrong, none of your degrees are soil science on their label.
I think my master's is says Masters of Forest Soils, I think.
Oh, okay, cool.
So how did you end up getting into this field?
Yeah, so I knew, in high school that I wanted to study soils. So I started with biology and chemistry, and took some geology. And then in grad school, I went to a forestry school, but worked with a soil scientist there and did soils research for my master's. And then my PhD was on how soils interact with invasive plants and how we can use soils to manage plant invasions.
So you must like digging holes. Every soil scientist I know spends a lot of time digging holes.
Yeah,
has like a favorite shovel.
I think my parents saw it coming because they would find me out in a hole in the backyard.
They call you Digger? Was that one of your nicknames growing up?
It could be a new nickname, Digger.
That's not very far from your son's name or nickname. Isn't that true?
He's Otter. My son is Otter. Yeah.
Otter and Digger. You guys, yeah, make quite a pair. Andrew, I've only taken one real soil science class in my training so far, but my teacher, I remember very clearly, said dirt is soil out of place, and so what exactly is soil, and how is it different from dirt?
So dirt is pretty much, if you've got your fill run from if you're doing construction, like that's dirt. If it's basically just mineral soil that hasn't been affected by plants and soil organisms, if it's just that primary crushed material, so primary rocks that have been crushed down, that's dirt. But soil is sort of an ecosystem of interacting plants and soil organisms. So it's a living system.
How many organisms would you say are in just like a tiny pinch of soil?
Billions. Yeah,
Wow.
so there will be billions of bacteria, archaea, fungi, protists, water bears, nematodes. Yeah, so billions, but it's mostly bacteria. They're tiny, tiny, tiny. I think maybe a more useful way to think about it is what's the biomass. So it'll be common to have, say, 300 grams of biomass per meter squared. So that would be like 3,500 pounds per acre of microbial biomass. So 10 times higher than your cattle stocking rate.
When you say biomass, Andrew, you mean like live stuff that's alive or was once alive?
Living bacteria and fungi and nematodes, all those soil organisms.
So dirt is soil that's dead, is what I'm hearing, and that difference.
Yeah, pretty much.
Yeah. All right. There's a thing about layering of soils. Can you talk about that, please?
Yeah, so you can tell a lot about the history of a site from looking at the layers of the soil. And what the layers are, generally at the top you have a dark layer, and that's because there's a lot of organic material, and that's because leaf litter from plants lands on the surface and decomposes and percolates organic carbon down into the soil. And then the fine roots turn over all the time. And so they're growing and dying, and it's all carbon that makes the top dark. And then depending on how productive the site is, the soils will be darker and deeper. So, around here, in some of the drier areas, you might just have a few inches of dark layer. Around here with grasses, we might have a foot of dark carbon layer. So that how big and how dark that layer is tells you a lot about how productive the site is and the climate of the site. And then below those layers, you get lighter colors. And depending on where you are and what the climate is, you can get the decomposition of the minerals at the surface. And so they move clay particles down through the soil. So you can get different layers of like a caliche layer, so like a calcium carbonate white layer that tells you how deep water percolates and then evaporates. You can get similarly, you can get like aluminum oxides, you can get clays down in a clay layer down in the soil, and that tells you sort of how much erosion there's been from the soils above. So then that's a function of the amount of water and temperatures that you have. So those layers tell you about the sort of the climate and history of the site.
So you said the darker layers are the more productive soils and happen in our grasslands. What are the more productive soils? What place in Utah has the most productive soils?
So it's a little bit of a tricky question because they all interact, right? So, but mollisols in general are considered very productive. And so these actually occur in kind of semi-arid areas that are dominated by grasses. Grasses have at least half of their biomass is put below ground. So they have a lot of root production, and they make those dense root mats to sort of store carbon, to harvest water, and to sort of put some carbon below ground so that they can regrow after they've been grazed or burned, so that have some insurance for regrowth. So mollisols, the whole Midwest of the U.S. is famous for having these really thick, deep, a foot of dark carbon soils. So those are highly productive soils.
In forestry, we don't talk very often about soils, but I hear when we talk about aspen, they're often associated with these mollisols. Can you talk about that?
Yes. So I think a good forestry example is, the classic hemlock hardwood comparison. Like in the Midwest, you can get areas where you can have a site, you could have an abandoned farm. By chance, hemlocks might start to grow, or by chance you might have hardwoods start to grow. And if you get hemlocks, they have a very acidic duff that decomposes slowly. And so they get a thick layer of leaf litter on top, and then kind of a poor soil below, and the nutrients cycle slowly. And then if you have a hardwood stand start, those leaves have a lot of calcium, they have a lot of nutrients, a lot of cations that buffer the acidity. And so they have a higher pH, they have a higher nutrient availability, have faster nitrogen cycling rates. And so there you don't have very much leaf litter on top, but you get a lot of nutrients moving down into the soil, and you get faster nutrient cycling rates. And so you could have started with the same sort of mineral soil, the same abandoned field, but you get two very different soil systems. And once the hardwoods and the fast nutrient cycling starts, then hardwoods can grow better there. And once the hemlock starts to grow, the slow nutrient cycling makes it hard for hardwoods to grow, and so the hemlock stays there. So once they get started to separate, they create these two different systems that stay in their own place. And so aspens are pretty similar. Like if you have aspen versus a spruce or Doug fir stand around here, it's a similar story. You get that more recalcitrant litter in the conifers that kind of sits on the surface more and slows nutrient cycling, versus the nutrient-rich, cation-rich aspen litter. The aspen litter aspens also let more light through, and they have more understory plants. And so you get more diversity and more litter production that decomposes quickly in those aspen stands.
Thank you.
So you had mentioned mollisols, and so I'm wondering if you could talk a bit more about soil orders and what soil orders are found here in Utah.
Mollisols are a common one. We have a lot. The dominant ones, though, are iridosols. So iridosols are just these dry soils. They're not very well developed. They don't have a very thick organic horizon because the production is pretty low. They may also have a caliche layer where the calcium carbonate moves down, shows you how deep water percolates. We also have a lot of inceptisols. If you think of like the sand dunes down south, or entasols, they're very young soils that just haven't haven't developed very much. And again, that's because there's not very much production. But yeah, we have alpha sols, inceptisols. We have, I think there might even be a few spodosols around, which are real conifer, acid conifer soils. But mostly we've got, mollisols and inceptisols are probably two of the most common.
So when you head down to Moab and everything is red, what makes the soil down there red? And then what order would you consider that to be?
Those are going to be, you'll have, if it's a really active area, those are going to be like inceptisols, really young soils. There's just not much, if you don't see much soil development. And yeah, so a lot of inceptisols, maybe some iridosols down there. The red comes from iron oxide. And basically you're seeing the red. I mean, we could have reddish soils here, but they'll look dark because there's a lot of organic matter covering all the red. But the iron oxides are pretty recalcitrant, and so they are hard to kind of erode away. But those sandstone soils are just silica oxide with maybe some iron and iron oxides in there, and those are very recalcitrant chemicals. They don't. It's very hard to decompose those further. So the reddish iron oxide remains, and there's not a lot of production, so there's not a lot of carbon to hide up that red iron oxide.
So I know that in that area, like when you go hiking on those trails and stuff, they have a lot of signs that talk about biological soil crust, and that the soil is alive and that there's things living on that top layer. Could you talk a little bit about what that is?
Yeah, they've been, Jane Belknap's group down in Moab has been doing it. USGS has been doing some really cool research down there. But so that crust is cyanobacteria is kind of the first part of that. They are a bacteria that can actually have a modal form. So they can sort of swim around through the soil when it wets up. And as they move, they leave kind of like a slime trail, like just like a slug. And that slime trail leaves all these sticky strings around that kind of hold soil particles together. Fungi do something similar. A lot of fungi, they exude glomalin, and that's why soils kind of stick together, is because there's these soil organisms that leave sticky trails around behind them. But so the biocrusts start with those actinomycetes, but then once they start growing, they're able to, or the cyanobacteria, they're able to fix some nitrogen, which is a limiting nutrient for plants. And so that increases nutrient cycling and plant growth, but then they also get some moss and fungi and other bacteria that will grow in there. So it's this web of threads that hold together the soil particles and fungi and lichen and cyanobacteria. And so it's this sort of living mat of soil organisms that cover the soil and kind of hold it in place. And it can be pretty, I mean, these are growing in rough environments where there's just not very many wet days in which organisms can grow. And so it can be slow to grow, but and to reestablish, it's kind of often hard for them to disperse and colonize new areas. And so once they're disturbed, you create again this feedback loop where once you've removed that biocrust, the soils can now erode more quickly. And once they can erode more quickly, it's harder for new biocrust to establish because it keeps eroding. If you think of a sand dune always moving across the landscape, it's hard for something to get to hold all that down. But the group down there, and I think at Northern Arizona University, they've been working on seeding, coming up with like a way to seed biocrust where they can put a little bit of inoculant, they put that cyanobacteria, they grow a little bit of it, and then they spread it out in areas that have been disturbed. And they've actually been able to get some reestablishment of biocrust pretty quickly.
So I've read and heard very different things about how long it takes for a biocrust to recover. I've read, you walk across it, and some research has shown as little as 7 years, and other things I've seen 100 years. What's your take on that?
Yeah, so a lot of that, depends on how much work you want to put in. If you seed it and go out and water it a few times, I think within a few years you could have a pretty healthy biocrust. But of course, you can't go water 10,000 acres in random places in southern Utah. So that's not very practicable. But I think you could, if you really worked at it, you could get a healthy biocrust in a few years. The problem is, the amount of water that you naturally get in those sites, the irrigation you put on, it would require naturally 10, 30, 100 years to get that amount of water to get that growth going. So it really depends on the site conditions, how much natural erosion there is, how steep the slope is, how much precip that site gets. So yeah, it's really going to be a function of how productive the specific site is, how wet it is down there.
And you talked about that. It sounded like a term when you talked about the web in the soil there, a term that I learned just a week ago, the soil food web. Is this the same thing?
No, that's a totally different web. So a food web is generally the idea that you have lots of different organisms feeding at sort of different levels. So I mean, we have food webs above ground. You've got hawks eating mice, eating seeds. So that there'd be a food web where that would be sort of a food chain, but then you get a web when you have organisms that are eating across different trophic levels. And so you get all these connections where, maybe changing a mouse might affect owls, which might affect coyotes, so there's the web means that you have these lots of interactions across different levels of organization. When we have the same thing in soils, soils are a pretty bizarre place though. A lot of the rules above ground don't really apply in soils. Like the whole idea of a species doesn't really apply below ground. So if you think about like a bacteria, the dominant organisms in the soils, and they can transmit plasmid DNA. So they'll just exchange DNA with each other. And so a bacteria may only live 3 hours, but in the end of its 3-hour life, it will have different genes in its body and it will have perform different functions. And so it's the idea of what is a species, and they just divide themselves. And so they're not really sexually reproducing. And so the whole idea of a species doesn't really work for soil bacteria.
How come there's never been a sci-fi movie made about this? Maybe there has. It sounds like a great storyline.
I think the trouble with tribbles.
So can you talk a bit about how soil organisms interact with plants?
Yeah, so I actually think of the plant soil atmosphere continuum. So I actually don't think of plants as a separate thing from the soil organisms. So 95% of plant families are mycorrhizal. So they have fungi that grow in and on their roots. So pretty much every plant, it has a very tight association with fungi. Some species, ectomycorrhizal species, a lot of tree species, the roots actually look totally different. They're stunted, stubby little roots because the fungi create a sheath around the root and really take over all the root functions for the plant. And those fungal cells grow in between the root plant root cells. They'll actually grow into the cell wall and sort of invaginate. So they're not quite in the cell membrane, but they're in the root well inside the roots. And so there's not really a clear distinction between the plant and the soil, right? So that fungal hyphal cell will grow in between the root cells, and then it grows out into the soil. And then in the soil, it exudes enzymes to decompose minerals and carbon from the soil. And so, the fungi is getting its nutrition by digesting things outside of its cells, and then it's moving that material through its hyphae and into the plant roots. And so it's very hard to draw a line as to where the plant ends and where the fungi begins and where the soil ends and the soil begins. So it's all this kind of fuzzy continuum. So yeah, I think it's very intimately connected, and we're learning more and more about crazy interactions between plants and just among all the soil organisms. So an example would be that there's a lot of bacteria that grow. They sense their environment, and when they find that they're in a good spot and they can grow quickly, they release these molecules, these quorum sensing molecules that tell all the other bacteria growing near them, this is good, let's go, 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 go. Like, this is great for us. And some of those bacteria plants don't like because they'll steal nutrients from them. And plants have learned to make anti-quorum sensing molecules. So they will pump out molecules that stop bacterial growth outside their roots. And so all these organisms are sort of tricking each other with chemical signals. And so they're all kind of talking to each other. There's a lot of bacteria that will release auxins, which is a plant growth hormone. So plants produce auxins to encourage their own root growth. When a plant finds a patch of water or nitrogen, they produce auxin that stimulates plant root growth. And so it encourages the plant to grow in good conditions. But there are some bacteria that have figured out how to do that too. So the bacteria will release auxin in order to stimulate root growth so the bacteria can eat that root. So they're all kind of talking to each other and tricking each other. It's a complicated web of interactions below ground.
Sounds like a tough neighborhood.
Yeah. I'm going to have a whole new perspective next time I'm digging my hands in my garden. I'm going to be like, whoa, what the heck's going on down here? And what am I interrupting? So you mentioned fungi, and you touched on mycorrhizae a bit. Is mycorrhizae found in all soils? Is that everywhere? Can you associate that with every plant that's growing, or is it only specific?
There may not be mycorrhizae in Antarctica. Depends. I think some of those northern tips have mycorrhizae. Pretty much mycorrhizae are everywhere. Almost all plants are associated with mycorrhizae. Pretty much any plant root you look at under a microscope, you will find mycorrhizae growing in the roots. So some notable exceptions from an agricultural perspective: the Brassicaceae, the mustards, they are amycorrhizal. So that's a big exception. Tends to be plants like the early successional ruderal plants, weedy plants, are less likely to be mycorrhizal because they tend to grow in high nutrient conditions. So you think of the plants that invade your abandoned fields, agricultural field, they succeed because they get to a place first, right? So if you're a plant that disperses widely and can grow in a disturbed site, then you're one of the few individuals there, and you have high resource availability because you're the only plant there. And so they don't want to associate with mycorrhizae and fungi to provide them nutrients because they've got plenty of water and nitrogen there because they're in an abandoned field. So those plants have evolved to prevent mycorrhizal infection and nitrogen fixing associations.
So I hear more and more about mycorrhizal applications in soils, and I hear both sides of it. Some people kind of think it's sort of snake oil and don't believe in it, and other people are all on board. Can you comment on that, please?
Yeah, I'd say invest in company. I think in 50 years, every crop, every restoration effort will be adding microbial inoculants. People are doing it now, and it's not too effective. It's a pretty blunt tool. I think there's a lot of failures out there. The idea is right. The execution is not well developed. So you can buy a lot of inocula. There are a lot of farmers using it. It is often not at all effective. I think we have a lot to learn about when those inocula are effective and where they're coming from and the specific interactions that are occurring. Because soils are, thousands of interacting sort of what we call species, just because they're 97% genetically similar. There's so many interacting species, we don't really have an idea of, can we add one fungus and change anything in the soil, or do we have to add a fungus and a bacteria, or do we have to add 100 together? And do we have to add a gram per kilogram, or do we have to add 1,000 grams per kilogram? We don't really know how to apply this stuff or the conditions under which they'll be successful. So we have a lot to learn about making it work. But in general, I would say as a rule of thumb, you can get soil organisms to increase plant productivity 25%. And so there's a lot of potential to use these inocula, but we're not quite there yet. It's worth giving it a try. You could certainly have big effects, and it can be effective, but it's going to fail a lot because we don't really know how to use it.
Is this part of your field of study, Andrew?
Yeah, I work on developing the sort of the concepts and understanding of how we're going to be able to develop those inocula and control plant microbe interactions.
So if a young person in our audience was wanting to pursue a field this way, they might come study with you, or are there leaders in this field you might point them towards?
Just look into soil ecology. Interestingly, the Netherlands is super into this. Wageningen University, I think the tiny little country of the Netherlands, it probably invests more in soil ecology than the United States or any other country. They're super into soils. They love it. It's like just a culture of soils there. So there's a lot of the best researchers in the world are there. This is what a lot of soil ecologists do, is trying to dissect and understand these interactions among soil organisms and plants.
So have you been to the Netherlands and studied soil?
I have not gone. I've worked a bunch with the people there, but I have not visited. I've met with them at conferences a lot and written papers with them. But
So could you please define inocula for us who may not know?
So yeah, inocula, there might be some official definition, but I just think of it as any organism you're going to add to a system. So anytime you cultivate, you grow, like in a petri dish, you can grow some bacteria or some fungi. Then you have all those organisms, spores or little parts of fungi or encysted bacteria, whatever. You have some of those that you've isolated or grown, and then you just add it to another soil. That what you're adding is an inoculation of those soil organisms.
Thank you. Sometimes we just need those very basic definitions. I certainly was hopefully not the only person who was thinking that in their mind, because I was like, what the heck does that mean? So you had mentioned the Netherlands. I wonder, is there somewhere, if you could study soil interactions anywhere on the entire planet or another planet, where would you choose?
Yeah, whether there are soil organisms on other planets is a big question. Like we've found bacteria in some of the deepest mines in the world in South Africa. I forget, they're like a mile or two underground, and there's no organic material down there. And the soil organisms deep underground are living because there's radioactive material, just natural radioactive materials underground. And those radioactive materials are splitting water. And the split water makes hydrogen, and hydrogen is a high energy resource that bacteria are living off of 1.5 miles underground. And so, and these organisms have been isolated from any evolution on Earth for probably almost 1 billion years. So that makes you think that it's very possible that there are similar soil organisms living on other planets. Where I would like to work, I will say that I do a lot of work on plant soil feedbacks, which is kind of studying how bacteria and fungi, archaea interact with plants. But most of that work is in North America and Europe, kind of temperate grasslands, just because it's easier to work in those systems, and that's where a lot of humans and universities are. There's very little been done in Arctic systems. There's been I think, rather than one site, I think there's a need to really look for patterns around the world. So is there a latitudinal pattern? Are plant soil feedbacks more important in Arctic systems or in temperate or in tropical? So I think getting more the bigger picture patterns of plant soil interactions would be something I'd be interested in doing.
That sounds exciting.
And a lot of exciting travel too.
Yeah.
Yeah, looking forward to that. Let's go down that tangent a little bit. Our audience doesn't know that Andrew's a really highly accomplished athlete and has biked the Continental Divide race and nearly won it several times, all the way from Canada to Mexico. And his list of accomplishments that way is quite long. And when you're out riding your bike or out on your skis, or probably harder on skis, are you always looking at soils, and can you sort of separate the two? Or
I think, well, a real advantage of these races is the training and the races. You just spend a lot of time by yourself outside. And so I think you just get a lot of time to absorb it. You're just sitting there on a saddle for 12 hours. You get a lot of time to let ideas pass through your head. So I think that's probably a big part of it. But I think, once you've dug pits, and I think it's kind of like you hear this with people who take their first intro to geology class. You'll never look at a road cut the same way, right? So I never don't look at a road cut. I'm on these little Forest Service roads all over the place. I mean, I always look at the road cuts. You always want to see what's going on underground. It's amazing. I remember, when you have to dig into soil, it's a lot of work. It's hard to do. And I remember for my master's, I just took endless soil cores and dug soil pits. And I remember walking into sites, and sometimes I'd get to a site and I'd see just the ground is just covered with rocks, and I'd be like, oh, this is going to be a nightmare. But I actually learned that you never know what's going on below ground when you look at the surface. There are some sites that just looks like flat, sandy, you're like, oh, this is going to be easy. And you can't get your soil core in 2 inches because it's just all rocks underneath the sand. And there are other sites where what happens is you get this freeze-thaw cycles, and so it pushes all the rocks to the top. And so you see all these like little baby heads, rocks the size of my hand sitting on top of the surface. But you push the soil core in and it's all smooth soil underneath, and it's really easy. So you never know until you dig a hole, you never know what's down there. And so, yeah, road cuts are a real treat.
I was recently paddling on Hiram Reservoir where they had older cuts, and I was just noticing a sagebrush was right on the edge, and its roots were exposed at 8 feet down. I wished I had my camera to take a photo of it.
Yeah, so another line of research I do, I use tracers, where I inject tracers in the ground to see where plants are absorbing resources. And one thing I've learned is that any plant can make deep roots, but they don't use them very often. So cheatgrass can grow roots down to 1 meter in just a few weeks, like in the field. So it's not hard for plants to grow deep roots. But around here, we only get 12 inches, 14 inches of rain a year. And so the rain doesn't usually go more than 1 or 2 feet below ground. And so it's uncommon for, if you grow a root to 4 feet, it's just going to sit in dry soil and not really do much for you. So those deep roots, plants can grow deep roots. They just usually choose to use shallow roots. I mean, precipitation comes from the ground, from the top down, and so most of that water is near the surface. And then all that organic matter, that really good soil, is right near the surface. So there's a lot of reason for plants to keep shallow roots. One interesting thing we found, I was working in Kruger Park in South Africa, and we planted some seedlings. So these were 6-month seedlings. They had like 4 or 5 inch roots from our pots. We planted them, and then like a month later, we measured where they were taking up their resources. Adult trees, grasses relied on the top 20 centimeters. Adult trees, like 10 meter tall, 15 meter tall trees, use the soil right below the grasses. So all the plants growing out there were in the top 20, 30 centimeters. The saplings that we planted were using soil water at like 50, 60 centimeters. So within a month, the saplings were kind of trying to avoid competition with all these old established plants, and they were putting their roots down deep and trying to use any resources they could down there. So yeah, most plants though try to keep their roots, most of the resources are near the surface, so most roots are pretty shallow.
You explained something much better than I do, a very common question for me. My tree is dying in my yard, from people all over Utah. And I always ask, it can almost always point to water and deep watering of trees. Because like you said, that grass is absorbing everything in the top, and you got to water past it to get to the tree roots.
Yeah,
So
but not too deep.
We have a bunch of registrant questions, so I want to get to those. So I hate to like cut us off, but I know that these people want their questions answered. So we're going to jump into these. Like a rapid fire style would be nice, so we can try and get through as many as possible. But if you need to take a moment to put a little bit more information on the answer, that's totally fine. I'm going to start with one that John Martin, who is here today, submitted. Will taking clumps of soil from a 200-year-old Pacific Northwest cedar and putting some into the soil of a 50-year-old cedar help that cedar in any significant way?
There's a lot of art to this science. Yeah, pretty hard to say. I would say you're 50-50 as to whether it would help or not. It's really, there's a lot of random conditions that determine. One thing I will say, I think that is a common misconception, is that mycorrhizae, if you have a high resource nutrient condition, so for example, if you have a potted tree in your yard in the city where there's a lot of nitrogen deposition anyway, or you're fertilizing and watering, mycorrhizae actually won't really probably help your plant. So mycorrhizae provide phosphorus and nitrogen and maybe water, but plants under high resource conditions actually won't let mycorrhizae infect their roots. So if you're already fertilizing, the mycorrhizae may not help that much. They can help prevent some infection, so they can kind of ensheath the roots and help prevent some infection or help exclude some pollution from the roots, so they may help in that way. But in general, under high resource conditions, plants will limit their mycorrhizal infection. So yeah, as far as how the microbial community develops over very old versus younger plants, there's not too much known about that. But you can try the experiment and let me know. Let me know what happens. I would say you're 50-50 as to whether, pretty random. It wouldn't be much more different than switching between two individuals.
Thank you.
Can you test for mycorrhizal? Is there a test to say if your soil has healthy mycorrhizal?
They're pretty much everywhere. It's more a function of exactly which ones they are. Like I said, mycorrhizae can be saprophytic. They can actually extract resources. They can be bad for a plant. In the same individual, mycorrhizae on the same individual plant, it might be good in some years and bad in others, depending on what the soil conditions are. So often conditions are stressful and the mycorrhizae provide water and phosphorus, but if you fertilize and the plant has all the water and phosphorus it needs, then the mycorrhizae is just going to steal carbon from your plant. So it's not, it's not as clear cut as we thought 20 years ago.
Can you touch briefly on the impact of climate change on soils?
On soils. So one thing is that we've increased the carbon dioxide concentrations in the atmosphere from, I think, 300 to 400 parts per million. Soils, because roots are respiring just like we do underground. They're not like the above ground plant parts. So carbon dioxide concentrations are usually 1,000 parts per million soil. So the CO2 concentrations don't really have any effect on soils. They're already way, way higher than our atmosphere. The CO2 is a fertilization effect for plants. It's probably increasing root exudation and carbon production. So it's probably the CO2 increases in the atmosphere are probably increasing carbon deposition into the soils. The flip side of that is that this higher CO2 concentrations, higher methane, higher temperatures increase respiration. And so it's kind of a wash as to the net effect. But in general, increased CO2 increases plant growth, which increases carbon sequestration in the soil, but higher temperatures result in more respiration. So it's kind of just accelerating the system.
Thank you.
We've talked a lot about mycorrhiza. Let's touch one more thing about that. How well do mycorrhiza buffer salts in the soil?
Yeah, there has been some interesting research using Inocula to help protect plants. So again, there's different types of mycorrhizae, like ectomycorrhizal versus endo, and so they behave a little bit differently. But in general, the mycorrhizae can deliver sort of a filtered water and nutrients, phosphorus particular, and nitrogen to plants. And so they can actually help filter out saline water and protect plants. So mycorrhizae are important, and most plants are reliant on them, but there are also a lot of bacteria that grow in sheaths around roots, and that can actually stimulate growth and protect growth in a similar way. So the roots, people talk about the rhizosphere because the roots exude sugars that feed organisms, bacteria around them. And so the roots are trying to get this sheath of bacteria and fungi growing around them to protect them from predators and from herbivores and from toxins in the soil. So yeah, there's a lot of potential to develop those.
So Brad Washa would like to know if you have found a connection between soils and cheatgrass. Or what have you found in the connection? I may have worded that wrong.
Yeah, so sort of the bottom line of what I found with cheatgrass, poa bulbosa, all the invasive species, they tend to, non-native species that have become invasive tend to benefit from soil organisms because they have been brought in from another continent, and all of the bacteria and fungi that have evolved to eat those plants are left behind, and then they show up in a new continent and they're like, oh, this is great, there's nothing eating me here, and so they explode, they take off, they do really well. Whereas the natives here they get eaten by the bacteria and fungi that are growing in the soil, and so they have a disadvantage. And so I developed this technique where we add activated carbon to the soil, and so that shuts down all the communication between bacteria and fungi and plants. And when I did that, the native plants lost their predators, and the invasive plants lost the benefit that they get from some mycorrhizae and growth promoting bacteria. And so it kind of leveled the playing field. And so it was able to increase, like blue bunch wheatgrass in particular took off, and cheatgrass died back, because cheatgrass in particular increases nitrogen cycling. It increases the growth and production of bacteria that turn over nitrogen. And so by adding the activated carbon, we slowed that nitrogen cycling, slowed cheatgrass growth, and increased blue bunch wheatgrass growth.
One of our viewers asked a question I'm really interested to hear your response on. Another question I'm interested to hear your response on. Jeremy Black asked, what are your thoughts on large burn pile restoration practices like you see on Forest Service harvest areas? And if you could perform the restoration, how would you go about it? The methods, rehab to prevent invasives and promote better natural vegetation.
Yeah, so carbon, I think, is really a key for soils, right? So a rich soil, think of as a dark soil, and that darkness is carbon. And so anytime we can add and spread carbon into the soil, I think is going to help. It helps for water retention. It helps for sort of binding nutrients. So it's anytime you get a burn pile, anytime you can get some carbon from that, and it can still be reduced form, or it can be in sort of the charcoal or activated form. But anytime you can add carbon to the soil, I think you're gonna help. I mean, the flip side of that is once you till a soil, once you've disturbed it, you've broken those hyphal networks, you release about 50% of the carbon in the soil within a few years. And that turns out to be terrible. So tilling, whenever you rip up the soil, you release a lot of carbon, and you very quickly reduce the soil quality. There's a cool study just from a couple of years ago showing a pretty direct correlation between soil carbon, soil microbial biomass, and wheat production. So the more carbon you have, the more microbial biomass you have, and the more wheat production you have.
So it seems here in our area, there's a lot of, probably over the entire state of Utah, people are just popping up houses everywhere. So they remove all of that topsoil, and then you're just kind of left with dirt and clay. What would you recommend as the most effective strategy to convert that into a productive soil?
Yeah, again, the key is carbon. Get some topsoil on there, get some carbon on there, mulching. So mulching is the carbon. One interesting thing is that we have a lot of limestones here. So a typical problem when you expose mineral soil is if it's a granite, if it's a silicaceous parent material, then you can get you'll develop some very acid soils, which really slows microbial growth, particularly bacteria. The fungi actually do a little bit better on acid soils. So you'll increase your fungal abundance, but decrease your total microbial abundance. But around here, when you have a lot of calcareous parent material, we actually don't have that acid problem because it's full of calcium and magnesium. So with our basic parent materials, that's not so much of a problem. But when you have that fill material, when it's that below that dark soil, there's just not much organic matter in there. And so those soils don't hold on to nutrients very well. And so the key is getting some organic matter into the soil. It's basically like a nutrient sponge. If you can add carbon, you've got a nutrient sponge that holds on to, sort of retains the nutrients and the water, which allows plants to get growing better.
Sort of answered this a little bit, but I'd be remiss if I didn't ask directly, especially since I see several people are asking about biochar, and that was our last topic. And what's your take on it? Do you think it's beneficial for soils? And please explain the difference between that product and activated carbon.
Yeah, biochar, activated carbon is basically clean biochar. So biochar still has a bunch of nitrogen and magnesium, calcium, phosphorus, has a bunch of nutrients left in it that haven't been totally cleaned out. Activated carbon has just been processed to remove everything except for the carbon skeleton. And so activated carbon is just a carbon skeleton. It will bind any organic molecule sort of indiscriminately because through van der Waals forces, it just holds on to organics. But it doesn't provide nutrients. And so it has a pretty different function than biochar. Biochar will provide some nutrients as well as bind organics. But again, like I said, carbon really seems to be the sponge, the foundation for good soils. And so if you can mix biochar into your soil, if you can add biochar, I think you're going to improve your soil quality. You're going to improve your water holding retention. You're going to improve. So the biochar is also, it is literally a sponge. It's just the matrix. It's the lignin tubes from the wood that's left behind. And so it's just these tiny, tremendous surface area. And when plants exude simple sugars out of their roots, those sugars get stuck into those tubes. And so it's just a place for bacteria to kind of hide out, prevent, or they can hide from grazing from amoeba and stuff. And they can grow and they can sort of keep nutrient cycling going, hold on to those sugars, keep nutrients available, hold on to water. And so it just improves your soil quality. So yeah, anytime you can incorporate carbon into your soils, I think you're gonna do better.
That was awesome. I've tried to explain this as a simple forester for years, and I've never come close to that. Thank you very much.
So we've got somebody who's asking about compost tea. So first of all, I don't have any idea what compost tea is. So if you could define that, but then could you also address if you believe repetitive compost tea applications in residential sites are effective?
Yeah, I think, so a compost tea basically it's kind of like, yeah, you're pulling the water out of compost. So you're basically cultivating, it's basically developing an inocula. So I would say you're also adding a lot of carbon. So you're doing well there, and you're growing these soil organisms that decompose these hard to decompose materials. And so you're just adding a lot of microbial inocula and biomass. In general, what happens with the tea is that by growing things with the compost material, you don't have living plants in there. And so you're cultivating all these soil organisms that cycle nutrients, but are not plant pathogens, right? Because there's no plants growing in that compost. And so you're creating this inoculate that's full of organisms that turn over nutrients and do not include organisms that feed on plants. And so, yeah, you're most likely to be adding organisms that are going to help plant growth. And so, yeah, it's great. I think it's a great idea. It's a great way to get beneficial organisms into the soil.
Thank you.
Got to visit a local composting operation here recently, and they talked about all the different products that they're making, and they kind of talked about this group of additives they called sizzle, just things that you can add to the soil, essentially to sell it. It was their attitude or point of view. And one of our viewers asks, are there snake oil soil enhancers on the market that we should be wary of? What's your opinion on, you've already talked about mycorrhiza, but humates, and he talks about organics. I'm not familiar with that.
Yeah, I'd say most of them are snake oil. So it'd be my default answer. But I will say there's a lot of potential. The theory is, I think, correct. And I think the application is going to increase and become more successful. I'm a little leery that there's a lot of people trying them and they're going to fail most of the time. And I'm a little worried that it's going to burn out the market for them because they're not very well developed or tested and often not successful, and because we don't understand really the conditions under which they will be successful. So we need to figure out how those are going to work a bit better. Right now, what happens is somebody says, oh, we found a mycorrhizae that increases, whatever, our seedling growth by 25%. And it worked here in North Carolina. And so we're going to sell it all over the world. And you're like, oh, but it turns out North Carolina soils are quite a bit different than Washington State soils, and it doesn't work at all in Washington State. So, yeah, so we have to learn the conditions and locations and species it works with. So there's a lot of potential. That said, I mean, I think it's worth doing some tests. I think, if you can spend a few $100 and try it on a few of your plots, you might find that in your site, oh, it's fantastic and it could work. I wouldn't be super optimistic about it. I think it's worth playing with and experimenting with. And I think in 20, 50 years, we're all going to be doing it.
So that bag of humate that I buy at the local store, I'm not going to say its name, should I not be putting that on my lawn? Is it really just doing nothing but taking my money?
Well, the humate is a little different. I would go with your own. I'd go with your own results. The humate, I think, is a little more promising, just because you're adding carbon again to the soil.
Thank you. So, Darren, you want to pick our last question? We've got 4 minutes.
Well, I'm going to put you on the spot. Maybe I've never heard of this. It's maybe one of the more unusual questions. Jumping worms and soil damage. Does that mean anything to you?
Yeah, so it's interesting. So we do have native earthworms in North America, but we don't. So the glaciers removed all earthworms from North America. So, in the northern U.S., there are no native earthworms. There are nematodes and roundworms, small worms. So those areas for 10,000 years have not had earthworms. The earthworms that we see are all introduced, typically from other parts of the world. The southern U.S., it's a little different. We did have native earthworms. But so in general, earthworms, they can turn over a lot of material. I remember being in sites in Connecticut where I went to grad school, and there would be areas of beech forest that the ground was just pure earthworm castings. Like there's just 100%. There's no leaf litter. It's just earthworm castings. So they can really turn over the soil. So they really increase nutrient cycling. They can both increase infiltration, water percolation, and decrease it. Those castings in some spots actually decrease percolation because they've removed all the leaf litter. They can increase erosion. So kind of depends on your previous site conditions. If you had really compacted soils, they might help. But if you had a forest, like if you had a healthy forest with a nice litter layer, that actually they can hurt because you've removed that litter layer and now you've got dense soil exposed to the rain. So it's a little bit complicated, but jumping worms are another non-native earthworm that have been introduced. Pretty much any big earthworm you see is going to be not native. The native earthworms we had were pretty tiny. So they're invasive. They increase nutrient cycling, turnover. They chow through a lot of soil material and turn it over.
But do they jump?
They wiggle quick. And that's why they're called jumping worms. You can tell them they have a very white, very light colored clitellum, that little white strip. That's how you can tell them from the other ones.
I'm picturing the movie Tremors.
Yeah, they're a bit smaller than that.
Okay, so we're going to wrap up with just two little simple questions. What is your favorite part about soil science, and then your least favorite part?
The favorite part is certainly that we have so much to learn. Like, I mean, it's just such an open field. Like, it's pretty exciting. Like, we don't know basics. Like, one of my basic lines of research is where do plants get their water from? We don't know, because it's hard to dig a hole, and when you've dug a hole, you've killed everything you want to look at. So we can still get some very fundamental, pretty cool answers that we don't have yet. My least favorite is that still not enough funding and respect for soils.
Well, thank you so much. I opened up a poll very late because I was way too invested in all of this interesting stuff. So if you're still here, we would really appreciate it if you take a moment to fill this out. Thank you so much for being with us today, Andrew. I learned a lot. Soil is just beyond fascinating. You know, I guess when I'm planting things, I don't really think about it, but then when you sit down and talk about it, you're like, wow, there really is a whole nother world down there. And so it's really fascinating to learn about that. If anybody had a question that they didn't get answered, feel free to email me and I will try and get that question answered for you. But yes, thanks again so much, Andrew. We really, really appreciate you being here today.
Thanks for having me. It was fun.
Thank you.
Bye, guys. Have a great weekend.
Good job back, Gabrielle. Thank you. Bye-bye.
Thanks, guys. That was great.